In this video, I want to explain the difference between human cells and bacterial cells. And one key difference is that human cells are eukaryotic cells, while bacterial cells are prokaryotic cells. Another key difference is the size. Human cells are relatively larger than bacterial cells. But I'm going to draw both cells the same size just so we can show what's going on in these bacterial cells. But what are some differences between these human cells and bacterial cells? Well, just looking at them, we notice we have different genomes. Humans have these linear chromosomes, and again, we have 23 pairs of these linear chromosomes, while bacteria just have one strand of a chromosome, and really it's circular, but it won't always be perfectly circular, but that's the difference. Humans have linear chromosomes, while bacteria have circular chromosomes, one-stranded circular chromosomes. So what are some other differences? Well, we know humans, we have this plasma membrane, and we know this plasma membrane is made out of these phospholipid bilayer membranes, but we also have other membrane-bound organelles. For example, our nucleus and our mitochondria and lysosomes. We see these are membrane-bound organelles made out of these phospholipid bilayer membranes. So we have these compartments. We have these enclosed membrane-bound compartments. And again, with our nucleus and our endoplasmic reticulum and Golgi and peroxisomes, etc. However, bacteria are very different. Bacteria also have this plasma membrane, which is also a phospholipid bilayer membrane, but that's it. Then inside the cytosol is just an empty space. There's no enclosed compartments with these phospholipid bilayer membranes. We just have a plasma membrane and then this empty space with our genome. We don't even have a nucleus. These bacteria are just completely open. So that's one key difference. Another key difference is, again, humans have this phospholipid bilayer membrane, this plasma membrane, and that's it. That's the barrier between intracellular and extracellular. However, bacteria are different. They also have this membrane, this phospholipid bilayer membrane, but they also have a cell wall. So bacteria have cell walls. Humans don't have cell walls, but bacteria do. This Peptidoglycan cell wall. And this cell wall is made out of, again, this Peptidoglycan. That's what the cell wall is made out of. And it's made out of these parallel polysaccharides that are covalently linked to these peptides. So we have this covalent structure forming our cell wall. And it's a little more complicating because there are different types of bacteria. Certain bacteria have this plasma membrane and this cell wall, but other bacteria have a inner plasma membrane, a cell wall, and an outer plasma membrane. So this would represent gram-negative bacteria, while this would represent gram-positive bacteria. So there's a little diversity. But we see the differences essentially between the different types of bacteria and humans. And another key difference is, let's say we're human and we have our genome. We know we can take our genes to create proteins. And we know the way we do this. First, we take our genome, then we transcribe it. We transcribe it into mRNA. Then we take the mRNA and we do some processing. We process the mRNA. Now the mRNA can leave the nucleus. And when it leaves the nucleus, it can run into these ribosomes because we have these ribosomes in the cytoplasm. And now once the processed mRNA runs into these ribosomes, we know now the mRNA is going to get translated. It's going to get translated, creating this nascent polypeptide. So again, we continue translating it. And once we fully translated it, we've made this peptide. And now the peptide breaks off. And that's how humans create proteins. That's how humans express our genes to create proteins. But bacteria are a little different. So again, we know bacteria, we have this linear chromosome, but I'm just focusing on a part of it. So it's the same idea. We take our the bacteria take their genome and they transcribe it into mRNA. So it's in the process of transcribing this mRNA, and as it's transcribing this mRNA from 5' to 3', the 5' part breaks off. But then again, we know we continue transcribing, making more mRNA. But now this is where it's different. Remember, we don't have a nucleus. We just have this empty space, and in this empty space, there are ribosomes. So as soon as we start transcribing this mRNA, it's going to run into these ribosomes, which is going to start to translate it. So notice this is a concerted process. We're transcribing mRNA and translating the mRNA at the same time. As once this mRNA is transcribed, it immediately starts to get translated. And that's different from humans. Humans first take the genome, then sequentially, then it makes the mRNA, which has to leave to run into these ribosomes to get translated. But bacteria, it all happens at the same time. So now we continue transcribing, making more mRNA, and we continue translating. Then we continue this process, and then eventually this bacteria has fully made a peptide. So notice the differences. And something else that's important to realize that's different are humans use a certain type of ribosomes while bacteria use a different type of ribosomes. Humans have a 40S and 60S subunits while bacteria have 50S and 30S subunits. So human cells and bacterial cells use different types of ribosomes. So now let's imagine you, the viewer, let's imagine you get a very nasty bacterial infection. How do we cure that bacterial infection? Let's say you're really sick and you have a really nasty bacterial infection. How do we kill those bacteria in your body? Well, we could give you an antibiotic like tetracycline. So what tetracycline does is it selectively binds to the 30S subunit of bacterial ribosomes. However, it doesn't bind to human ribosomes because remember, we have different types of ribosomes. Humans have different ribosomes compared to bacteria. So this tetracycline selectively binds and inhibits bacterial ribosomes. So if you're sick with the bacterial infection, but you take this tetracycline, it's going to essentially bind, it's going to enter the bacteria in your body, bind to this 30S subunit and inhibit it. So now the bacteria in your body can't create proteins, now can't translate proteins because again, we're inhibiting the ribosomes and now the bacteria die, they can't make proteins. Another type of antibiotics are these penicillin molecules. So we know bacteria have enzymes to make their cell walls. And if a bacteria wants to divide and propagate, every time it divides, it needs to make a new cell wall. And to make these new cell walls, it needs enzymes. So this penicillin inhibits these cell wall making enzymes. And again, we can take penicillin and it'll have no effect on humans because humans don't have cell walls. So we don't have enzymes to make cell walls. However, bacteria have enzymes needed to make their cell walls. So if you take penicillin, it's going to inhibit those enzymes and now these bacteria can't divide. So that's how you get rid of the infection. Another type of antibiotic is this rifaximin. So essentially what this antibiotic does is we know humans, we have RNA polymerases to turn our genes into mRNA. But bacteria also have RNA polymerases to convert their genome into mRNA. However, bacteria RNA polymerases are different from human RNA polymerases. We use different RNA polymerases. So therefore, this antibiotic specifically and selectively inhibits bacteria RNA polymerases. So if you're sick with the nasty bacterial infection, but you take this antibiotic, it's going to inhibit the RNA polymerases of the bacteria in your body. So know that bacteria can't essentially express their genes. So now the bacteria die. So if you're trying to design and develop good antibiotics, you need to take advantage of the differences between human cells and bacteria cells. And you have to think of antibiotics that will selectively kill bacteria cells while leaving human cells fine. However, this can get complex. For example, let's say we have this species of bacteria. We know that this bacteria is going to rapidly divide. But we know while these bacteria cells divide, they're going to replicate their genes, and this replication process isn't perfect. So there's going to be a little bit diversity. So even though they're all the same species, every time they divide and replicate their DNA, it's not perfect. So they're going to develop some diversity, and they're all going to have slightly different genomes with different mutations and some diversity. So now let's say we have these bacteria, this population, let's say we give it tetracycline. What's going to happen? We know this tetracycline inhibits the ribosomes, so now these bacteria are going to die. But remember, they all had a little bit of diversity. Every time they replicate their genomes, they all have a little bit of diversity with their genes, with their mutations. So let's say just by chance, we take this tetracycline, it's going to kill most of these bacteria. But there's a little diversity. Let's say just by chance, these two bacteria happen to get mutations that made them resistant to this tetracycline. Maybe these two bacteria had mutations that gave them slightly different ribosomes, so now they're resistant to this tetracycline. So now what's going to happen? Now these bacteria that were resistant to the tetracycline are going to divide and create their new lineages. And now all these bacteria are resistant to tetracycline. So this is bad. This is antibiotic resistant. These bacteria are resistant to tetracycline. So this is bad. So what do we do? Well, now let's try another. Let's give them another antibiotic. Let's give them penicillin. What's going to happen? We know we're going to kill most of the bacteria, but let's say just by chance, this bacteria happened to have a mutation that made it have a different enzyme that made its cell wall. So maybe this bacteria is resistant to penicillin. Now what's going to happen? Now this bacteria is going to create its own new lineage, and now all these bacteria are resistant to penicillin. So what now? Well, maybe let's give a new antibiotic. So we give a new antibiotic, and it's the same idea. Most of the bacteria are going to die, but let's say just by chance, these bacteria with that diversity happen to have mutations that made them resistant to this antibiotic. Now these bacteria are going to divide, making their own new lineages, and now this is a problem. These bacteria are very dangerous. They're resistant to all of these antibiotics. We gave these bacteria chances to essentially mutate, form mutations, and form resistance. So now this lineage of bacteria are resistant to all these antibiotics. So this is terrible. This is dangerous. How do we deal with this problem? This is a huge problem. Well, let's take a completely different approach. Instead, let's say that these are different. Let's say this is a different person with a different infection, with maybe some different bacterial infection. So what do? Well, instead of giving it one antibiotic and giving them a chance to develop resistance, let's say at the same time, simultaneously, we give 3 different antibiotics all at the same time. What's going to happen? Well, we know even though they're all the same species, they're all going to have a little diversity. So maybe this bacteria, just by chance, happens to have a mutation that makes it resistant to tetracycline. However, what's the chance that this bacteria is going to have a mutation that makes it resistant to tetracycline, penicillin, and rifaximin all at the same time? That's very unlikely. It was actually very rare for a bacteria to develop resistance for a single antibiotic. So what's the chance that one bacteria in one generation will have enough mutations to make them resistant to all 3 antibiotics all at the same time? That's very unlikely. So if we give, once we get the infection, if we give 3 antibiotics all at the same time, then they're all going to die. It's very unlikely that there was one bacteria that all at the same time within one generation happened to have mutations that made it resistant to all these antibiotics at the same time. That's extremely unlikely. So that's how we deal with antibiotic resistance. You follow your regimen. If you get sick with a bacterial infection or some infection and your doctor prescribes you a very specific antibiotic regimen, listen and take it very seriously and carefully and follow.